On a sunny springtime stroll through a park, it is easy to ignore the parts of plants that are hidden from view. Plant biologists see things differently. They look below the surface where plant roots are organized in elaborate systems that are critical to the organism's development. Intricately organized tree root systems, for example, can span as far underground as the tree grows high above the soil. Applying an advanced imaging technology to plant roots, researchers at the University of California, San Diego and Stanford University have developed a new understanding of essential root chemicals that are responsible for plant growth. Using a type of mass spectrometer, a study led by UC San Diego Biological Sciences postdoctoral scholar Tao Sang and Assistant Professor Alexandra Dickinson produced a roadmap that profiles where key small molecules are distributed along stem cells of maize, corn plant roots, and how their placement factors into the plant's maturation. The findings were published in the journal Nature Communications. This chemical roadmap provides a resource that scientists can use to find new ways of regulating plant growth. The researchers adopted the technology called disruption electrospray ionization mass spectrometry imaging or DESI-MSI to probe plant roots for the chemicals involved in growth and energy production. They initially focused on maize plants at the root tips where stem cells play an active role in the plant's development. Their method involved cutting through the center of the root to get a clear image of the chemicals inside. The researcher says that their imaging system sprays out droplets that strike different portions of the root and dissolve chemicals at that location. A mass spectrometer collects the droplet splash and tells them what those dissolved chemicals are. By systematically scanning the droplet target spot, they make a spatial map of the root chemicals. The resulting images, believed to be some of the first to reveal the transition between stem cells and mature root tissue, show the foundational role of metabolites, molecules involved in the plant's energy production. Tricarboxylic acid (TCA) cycle metabolites became the focus of the research since they were found to be a key player in controlling root development. Coming into the study, the researchers expected a relatively uniform distribution of chemicals. Instead, with their chemical roadmap in hand, they found that TCA metabolites are clustered in patches across the root. The Dickinson lab showed that these TCA metabolites have predictable effects in development, not only in maize, but in another plant species as well, Arabidopsis. This is likely because TCA metabolites are highly conserved. They are made in all plants as well as animals. Also emerging from the new images were previously unidentified chemical compounds. Dickinson says the mystery compounds could be critical for plant growth since they also are grouped in patterns at specific locations, suggesting a prominent role in development. Dickinson and her colleagues are now investigating these compounds and comparing varieties of maize that have different levels of stress resistance for adverse threats such as severe climate conditions and drought. Their new information will help them develop novel chemical and genetic strategies for improving plant growth and stress resilience.